eventually assemble into a virus particle. One of the questions that's outstanding is how this viral assembly occurs and how much does this GAG protein interact with the membrane proteins of the cell, as well as the RNA, which is the genomic elements in the HIV virus. And so what Hirsch has been able to show is, together with collaborators at the NCNI, is that in the absence, so the question of how does the protein associate with the membrane, they mixed the protein with the tethered bilayers and they saw a conformation where the protein essentially collapsed, where both the matrix and the NC through electrostatic interactions will interact with the surface and create this kind of a structure. What they found, which actually is kind of interesting, is that the neutrons, the reflectometry, could not only detect that structure, but if a model nucleic acid, which is known to bind the NC and mimic genomic RNA binding to an NC, is added, the protein actually, the reflectivity profile changed dramatically, and the protein moves to this extended conformation, presumably bound by the RNA, so that the part of the molecule that was once associated the NC with the bilayer is now extended and bound to the RNA in this extended form. So it shows that and the structure of this protein is actually in a third state when it's free in solution. So it shows that the context, again, of the protein structure in the presence of the membrane is different from the solution. And then, in addition to potential ligand partners, you can see fairly dramatic conformational changes in these proteins on the surface. So another example from Hirsch's group is the P10 protein, which is basically involved in cell cycling and turning the cell cycle off. So it's a